Structure of proteins. Determination of structure of proteins. The different steps involved are: 1. Elemental analysis. Most of the protein contains carbon, oxygen, hydrogen and nitrogen, along with very small amount of sulfur and phosphorus. The second step is determination of molecular mass. And the molecular mass of proteins usually vary from about 10,000 to millions. And depending upon the complexity of the structure, the protein structure can be divided into 4 different levels. These are primary, secondary, tertiary and quaternary structure of a protein. Primary structure. The covalent structure of a protein is called its primary structure. It gives the sequence in which the amino acids are held in the polypeptide chain. And the primary structure is very important regarding the biological properties of proteins, because the change of a single amino acid can disrupt the biological property of the protein. For example, the disease sickle cell anemia results due to defective hemoglobin, that is, the replacement of a single amino acid in the protein hemoglobin. And the primary structure of a protein can be written like this. So the different amino acids are held together in the polypeptide chain through the amide linkage, that is CONH linkage. And in the polypeptide chain, there in one end there is a free amino group, and on the other extreme end of the polypeptide chain there is a free COOH group. And the amino acid corresponding to that is known as, for example, in this case, the amino acid having free COOH, that is known as the C-terminal amino acid, and the amino acid which is having free NH2 group on the other end, that is the N-terminal amino acid. Determination of primary structure of proteins. The various steps involved are hydrolysis. So by using acids, bases or enzymes, the polypeptide is broken into different amino acids. The second step involves separation of amino acid using chromatographic techniques or ion exchange method. And the third step is sequence of the arrangement of amino acid. And that is usually done by a combination of partial hydrolysis and the terminal residue analysis. The terminal residue analysis involves the determination of the C-terminal amino acid and the N-terminal amino acid. And for determining the C-terminal amino acid, there are two important methods. One is hydrosynolysis method, and in this method, the peptide is heated with anhydrous hydrazine when all amino acids except the one at the carboxyl end of the polypeptide are converted into the corresponding hydroxides. So if we consider the polypeptide chain, so consider a peptide having 3 amino acid. When this is heated with hydrazine, what happens is the bond breaks at this position, and the first 2 amino acid gives the corresponding hydroxides having the formula NH2 CHR1 CO NH NH2. This is the hydroxide of carboxylic acid 1 plus NH2 CHR2 CO NH2, that is hydroxide of second amino acid, along with the C-terminal amino acid in the free form. So NH2 CHR3 COOH. So by identifying the free amino acid, we get the nature of the C-terminal amino acid. In the carboxypeptidase method, for the determination of C-terminal amino acid, the polypeptide is treated with the enzyme carboxypeptidase, and this enzyme attacks the polypeptide chain only at the end, which contains the free carboxyl group. So when the terminal amino acid is liberated, the new terminal free carboxyl group will be attacked by the enzyme, and after a given time of hydrolysis, a number of terminal amino acids will be liberated from the polypeptide, and by their identification, the sequence in the peptide chain can be established. Next step is determination of N-terminal amino acid. One method is Sanger's method. In this method, 1-fluoro-2,4-dinitrobenzene, and that is Sanger's reagent, is taken, and this reagent reacts with the free NH2 group of the N-terminal amino acid to get the DNP derivative, dinitrophenyl derivative, along with free amino acid. So the DNP amino acid corresponds to the N-terminal amino acid. So the compound is 1-fluoro-2,4 dinitrobenzene, and that is fluoro. Dinitrobenzene reagent or Sanger's reagent. And when it is treated with a polypeptide, the N-terminal amino acid reacts with, so if we consider a polypeptide chain like this, so a peptide having 3 amino acid, when it is treated with sodium hydrogen carbonate, with Sanger's reagent, the free NH2 group combines with the fluorine and we get the DNP derivative having this structure. Which on hydrolysis we get the DNP derivative of the N-terminal amino acid along with the free amino acids. So by identifying the dinitro derivative, we can identify the N-terminal amino acid.